Good morning, everyone. It is Tuesday and January the 12th. It's 9 a.m. here in Cleveland, Eastern Standard Time. My name is Ashok Agarwal. I'm your host for the next 2-3 hours. And I want to, first of all, before we begin the program, want to wish you a very happy New Year 2021. I wish you good health and a lot of happiness, and I hope that we will be able to see many of you during this year after the pandemic is under control. So thank you again. And today is a very exciting program. We are here to discuss and present virtually our online ART training. Today is the third program in the series. We began in November. We presented first program in November, then second one in December, on December 8th. And today is our third program on January the 12th. Today's program is on Ancillary Semen Test. So let me begin without taking any further time and begin my introduction. Very briefly, this is my biography, a very brief biography, just to provide few highlights. I am in charge of the Andrology Center at Cleveland Clinic for the past 28 years. I'm the director of the American Center for Reproductive Medicine, which is the host of this online virtual training session. I'm a professor of surgery in urology at the Lerner College of Medicine at Case Western. I'm a staff in urology and in some other department. Our group is very well known in the area of male infertility, andrology, and human-assisted reproduction, and we are ranked as #1 in terms of our publications based on the Scopus, which is one of the premier service in terms of the literature in the medical field. Our group has published a very large number of books in the field of male infertility, ART, fertility preservation, DNA damage, and antioxidant. Our laboratory has trained over 3,000 scientists, clinicians, graduates from more than 100 countries. The organizers and speakers of this program have no conflict of interest. We have taken no sponsorship or asked any company for that. This program is made entirely possible by the support from our foundation, from our own center, and the webcasting support has been provided by our colleagues, David Reichling and David Wolfley. Course faculty have generously donated their time for this online training, and our program coordinators, who you will see in a minute, have graciously volunteered their valuable time to manage this program. Lastly, the names, images, and description of products or instruments that you may see during the lectures or which will be discussed during the presentations are not being endorsed. They are being used routinely in our program. So this is today's program. The learning objectives today is we have a very, very good program today, and you will see this is part of the semen analysis, and it is like a hidden gem. Most people will not know many of these tests that are basically done as part of semen analysis as indicated, So we are going to discuss about the leukocytospermia, or detection of white blood cell or infection, sperm vitality testing, anti-sperm antibody screening, special test for azoospermia, post-vasectomy screen, and screening for retrograde ejaculate. So I can tell you that today's program is by far going to be the very best. We have invested enormous amount of time in preparing for these lectures. They have been well researched, well prepared, and practiced dozens and dozens of time what you are going to see today. So for each lecture, we have spent almost 30-40 hours minimum in practice sessions, and each lecture has gone through at least 10 or 12 practice sessions with multiple people, and each content has been well researched through the literature with proper references, and to make sure everything is extremely accurate and simple for you to understand. So this is our program today in a diagram. As you can see, the last lecture is going to be by our clinician, who's going to really put it all together to explain what these tests really mean to you. Dr. Sharma spoke very nicely about a little known test called vitality test, and he explained the differences between vitality and viability. Two very important differences, one for diagnostic, one for therapeutic. And now we dive into the next topic, which is on anti-sperm antibodies. And our next speaker is Dr. Sejal Gupta. Dr. Gupta is a veteran of our andrology program. She is the technical supervisor of our center, which runs 3 laboratories in different parts of Cleveland Clinic, in different cities. And also we have a very large reproductive sperm and semen bank for cancer patients. Dr. Gupta has a distinguished career. She has done her fellowship in reproductive medicine with WHO, and then a research fellowship with our program. She has been highly decorated, as you can see. She has also done a master's in embryology from the Jones Institute. She is a very passionate andrologist who really takes care of our patients in the most compassionate manner. And today I invite her to speak about the issue of anti-sperm antibody test. So Sejal, you can present if you want.
Thank you very much, Dr. Agarwal, for the introduction, and welcome to all the participants. Good morning from Cleveland. And the presentation that I'm going to be making is on anti-sperm antibody test. Next slide. The learning objectives of this talk are indications for anti-sperm antibody testing, review the principle and protocol for the technique by which we do the anti-sperm antibody test, which is the mixed antiglobulin reaction, and in short, it's called the MAR test. Learn scoring, calculation, and reporting of the results by this test, and recognize the importance of quality control, quality assurance, and troubleshooting. Next slide, please. So this slide illustrates the indications for sperm MAR test. So the most important indication is sperm agglutination, which could be mild, moderate, or severe. And on the right side, you can see on the illustration the presence of sperm that are agglutinated to each other. The second is low total motility, low progressive motility, vasectomy reversal. Any disruption in the barrier of the blood testes barrier can result in formation of sperm antibodies. Scrotal injury, which again leads to disruption of the barrier, previous miscarriages, and poor IVF outcomes. Next slide. So the equipment and supplies that are needed for conducting this test are the bright field microscope using the 20x magnification, phase contrast, and with the green filter. The others are what are listed are the incubator and some other supplies. And finally, what is needed is a fixed cell sperm counting chamber and a humidified chamber in which the samples are incubated. Next slide. So the reagents listed are, and we have provided the illustrations on the right side. The first important reagent is the MAR test IgG kit. And this kit is illustrated as the first illustration, and it is labeled. So this has initially the suspension of the polystyrene latex beads, which are 2 micrometers in diameter, and these are coated with the human IgG. And then there is the MAR anti-serum, which is containing the monoclonal antibodies towards the fragment C of the human IgG. The second important reagent is the controls, which are the positive and the negative controls. And these are run in the assay to make sure that the assay is run correctly. The third illustration on the top is the IgA kit. So this IgA kit has the latex beads and the anti-serum in a single solution. And you can see our technologist at the bottom. She's performing the sperm MAR test. Next slide. What is the principle of the MAR test? Essentially, there are 3 immunoglobulin classes, IgA, IgG, and IgM. But in the semen sample, the main antibodies are IgA and IgG. And what leads to the antibody production is the autoantigenic properties of the spermatozoa. And on the right side, you see the illustration where the beads are attached to the spermatozoa and the beads are coated with the antibody to the IgA. And these will attach to the sperm-bound IgA antibodies. So basically, there is a bridging antibody which is connecting the beads to the spermatozoa. And the motile spermatozoa and the beads will form mixed agglutinates, and that's why it's called a mixed antiglobulin reaction. And it can be tested against both IgA and IgG antibodies. And the binding of the beads indicate the presence of IgA or IgG antibodies. Next slide. So here I'll show you a short video clip of how the test is performed, the IgG test. And you can see these are there's a humidified chamber. So the slide is labeled, the semen sample, 10 microliters is placed on the slide. Next, we place the beads from the IgG, 10 microliters, and then the semen and this beads are mixed well. Next, we take the anti-serum 10 microliters and add it to the mixture, and then mix the sperm sample, the beads, and the serum. And then a cover slip is placed on the slide. This slide is placed in the humidified chamber. We place a timer for 3 minutes. So once the incubation is done, the reaction has happened, then this slide will be examined on the phase contrast microscope. Next slide, please. So what is the significance of positive IgA and IgG antibodies, which are both tested in the semen sample? The IgA antibodies have greater clinical importance than IgG antibodies. Essentially, the beads will bind to the motile and immotile spermatozoa, which have the surface-bound antibodies. And this is illustrated in a very nice illustration on the right side, the binding of the beads to either the head or multiple sites of the spermatozoa where they are bound, and the percentage of motile spermatozoa with the bound beads is recorded. So the important thing to understand is just count the motile spermatozoa with the bound beads. Next slide. Here, what are the quality reagents? The quality control reagents are basically decomplemented patient serum, which is diluted in cell culture medium, and both positive control and negative control is provided with the kit. These are warm to room temperature before use, and the shelf life is about 18 months. Next slide, please. So here I've provided a step-by-step protocol of how the MAR IgG positive and negative controls are run. So the first step is to vortex both the controls well before use. And the second step is we use a known negative. Known negative means this donor sample. It's a normospermic subject, and his sample was already tested for antibody and was negative. And this sample, to this sample, we add modified human tubular fluid, twice the sample volume. And once we have added the HTF, this is mixed well, the step 4, and then this sample is spun for 10 minutes at 1600 RPM. And once the centrifugation is done, the fifth step is to remove the supernatant and leave some of the supernatant, about 100 microliters, to mix the pellet. And this last step is used, washed donor sample is used to test the negative and the positive controls. And this I will illustrate in a video clip in the next slide. Next slide, please. So here, the semen sample from the donor is there, and then modified HTF is added, twice the volume. This mixture is placed in the centrifuge for 10 minutes. The aspiration of the supernatant is done. The pellet is left undisturbed. And then the pellet is reconstituted with some more fresh modified HTF. And the concentration should not be higher than 50 million. So this is the positive control. 50 microliters of the positive control is added to the amber-colored vial. And then the donor sample is added 50 microliters. Same is done for the negative control. And then once the mixture is in the vial, these vials will be incubated for 1 hour for the reaction to happen. And then the actual testing will be done with the IgG beads as well as the IgG anti-serum to check whether the positive ran positive or the negative ran negative. Next slide. So to examine the sperm MAR test scoring, what are the basic principles? The latex beads bind to the spermatozoa with antibodies on their surface, and there could be few beads or multiple beads that may attach to the motile spermatozoa. So as a result of the binding of the beads, the motility is reduced, and especially when there are large clumps of beads binding to the spermatozoa. The sperm, which do not have the antibody coating, will swim freely between the beads. So we will count only motile spermatozoa, and a total of 100 is counted, the ones that are attached versus the ones where there is no bead attachment. The ultimate goal of this test is to evaluate the percentage of motile spermatozoa with the beads attached. Next slide, please. So going further, how do we read the results? We use the 20x objective, as you can appreciate in the illustration on the right, and use the phase contrast with the green filter. And then count 100 motile spermatozoa twice to assess the percentage of sperm binding to the beads. What you report is the average of the two values you have obtained, and the test is reported as positive if greater than or equal to 40% of the sperm have beads attached. And this is per the IgG kit, the manufacturer recommendations that any test where it is greater than or equal to 40% binding should be reported positive. Now, as per a publication from Human Reproduction from Lotti et al., they have reported a prevalence of MAR test positivity in the 5 to 15% of infertile males. So it is an important cause of infertility. Next slide, please. Here I have an illustration of a negative control sample. So you could play the video. And what I want to show is basically there are beads, clumps of beads, but the spermatozoa are predominantly swimming or moving freely, so they are not bound to the beads. And so we have the negative control run as a negative, and we know the test is performed accurately. Next slide, please. Oh, there was another one. Please go back. The positive control. So here you will appreciate the positive control. And what I want to show you here is the clump of beads and the spermatozoa that are attached in this clump, and they're attached either by the tail or by the head, but they are basically bound to the beads. And so this was a positive control. Reporting of results, negative results are reported if less than 40% of the sperm is bound to the beads. Positive results, how do we report and record? Positive if greater than equal to 40% of the sperm is bound to the beads. We report the person binding, and we report the area of bead attachment, whether the head, mid-piece, because if it's bound to the head, then the clinicians have to make a separate judgment call. And on the right, you can see and appreciate the different areas where the sperm is bound. The first one shows the binding to the head. The second shows to the tail. At the bottom is a negative result where there is sperm that is swimming and there's no binding. Next slide, please. So quality control is an important component of the anti-sperm antibody testing. And in our laboratory or other laboratories perform the proficiency testing. So this is an example of two analytes. We are sent two analytes, and the numbers are provided 17 and 18 by the College of American Pathologists. So the first analyte with the red box shows 4% binding. It is negative. The second analyte shows 87% binding, and it is positive. And then the negative and the positive controls results are also shown. The positive control, 97% positive binding. Negative control, there was 13% negative binding. So we know the results are accurate, and this is what we report as part of EQC. Next. So these are the results for both the analytes, and the intended response matched what our results were. So the lab is performing the test accurately and reporting the results well. Next slide. So here is a scenario for a negative ASA IgA test. And this was an IgA ASA, or the anti-sperm antibody test, performed for a sample with sperm concentration of 15 million, motility of 45%. How do we calculate the percent total binding? It's number of sperm with bound beads upon total number of sperms counted and reported as a percent. So when we examine under the microscope, what we saw was free total motile 75, bound total motile 25. So the percent total binding is 25%, and the result is ASA IgA test negative. Remember, the cutoff is greater than equal to 40%. Next slide. This is scenario 2 that is illustrating a semen sample with low motility. So this was a semen sample with 5% motility. And these are the patients where anti-sperm antibody is ordered by the physicians, and a concentration here was 2 million. When the sample was examined under the microscope, there were less than 100 motile sperm on the wet prep. Person binding cannot be reported due to very few motile sperm, and hence the result was not reported to the clinician because of very low motility. Next slide. So this is a recent study published in Human Reproduction, which examined the significance of positive MAR tests. So on the first panel, the bar diagram shows percent of men with MAR test positivity. They took a cutoff of greater than 50%, and this was examined in males of fertile couples versus male of infertile couples. And you can appreciate that there was significantly higher number of positivity in males of infertile couples. The second bar diagram shows comparison of positive MAR test and negative MAR test in men with history of epididymitis. Epididymitis is, again, there is a disruption of the blood testis barrier, and what they have reported is a higher percent of MAR test positivity in patients with epididymitis. So It can be positive in patients with orchitis as well as in patients with epidermitis. Next slide. So I would like to sum up with some take-home key points. MAR test is useful in the diagnosis of immunological infertility. It is important to test for both antibodies, IgA and IgG. And anti-sperm antibodies do play an important role in male infertility, as has been demonstrated by several publications. And proficiency testing, or EQC, is an important tool for reporting accurate anti-sperm antibody results. On the right, you see our andrological evaluation of male infertility. This has the protocol for the MAR test provided in it. Next slide, please. So this is our group in front of the Andrology Center. And I thank you all for your attention.
You can. Let me introduce our next speaker, which is Dr. Sharma. And Dr. Sharma is going to be speaking again. His topic today is on a special test for azoospermia. This is a hidden gem. I am sure that many of you may not be totally familiar with this kind of test. So as a clinician or a laboratorian or a reproductive professional, you will really enjoy a test which has been used and developed by our program many years ago. So Rakesh, you can begin the presentation.
Thank you, Dr. Agarwal. Today I will talk on the special test for azoospermia. Next slide, please. The learning objectives are when to perform this test, which is also called the nuclear fast picro indigo carmine test, or in short, NF-PIC staining test. How to rule out the sperm in the wet prep. Understand the principle of the cytospin involved before the slides are stained. Review the NF-PIC staining steps. Understand the quality control and the reporting of the results. And in the end, I will leave you with some take-home message. Next slide, please. The incidence of azoospermia is 10% in the infertile men. However, the semen analysis is unable to identify the samples which are azoospermic. Our group developed and published a special test called the NF-PIC test for azoospermia, and this was validated and published in 1998. The image on the left shows the steps involved, and these are: the sample is examined on a routine semen analysis. If we do not find any sperm on the neat sample, the sample is centrifuged and examined. If no sperm are seen, the subsequent steps will be followed. But if sperm are observed in the spun sample, then this is a case of cryptozoospermia or hidden sperm. When no sperm are seen on the spun sample, then we proceed for the special test, which is the NF-PIC staining, and this is preceded by the staining and the cytospin step. And when the slides are stained, we will either see the sperm or we will not see sperm, and this is how we will validate the azoospermia test. The image on the right illustrates the protocol for the azoospermic test, and this has been shared with all of you. Next slide, please. So before the smears can be prepared, they have to be processed using the cytospin, and cytospin is a benchtop special centrifuge. It has specially designed rotors and sample chambers. The cells are deposited on a very clear defined area on the glass slide, and the cytofunnel is used, and this cytofunnel has a filter card which actually absorbs the excess of the fluid so that the cells are flattened in a small area on the slide, and the rare spermatozoa can then be identified by this test. On to the right, you see the cytospin and the inside of the cytospin. Next slide, please. The equipment and material required is the cytospin centrifuge, the cytofunnel, and the microscope. Next slide, please. The steps involved in the cytospin are: the sample is mixed and the slide is placed on the slide holder. The cytofunnel is placed on the metal slide holder and a drop of semen sample is added. And this is followed by a drop of saline sample. Next slide, please. Here I would like to show you a short video clip, and the patient information is provided on the slide, and the slide is placed in the metal holder. You can see the small circle on the metal holder. That is where the slide sample will be followed by the cytofunnel. The cytofunnel is placed in the metal holder and placed in the rotor. The sample is well mixed and a drop of the sample is added onto the cytofunnel. We also include a blank slide which will not contain the semen sample but will only contain the saline, and this slide is used as a quality control. Once the samples are placed, these are capped. Here you can see we have added a drop of saline and the samples are capped. The rotor lid is placed and the sample is spun to allow the sample to be deposited on the slide. Next slide, please. The NF-PIC staining allows to identify the rare sperm when no sperm are seen during the initial semen analysis. And these spermatozoa can be identified using a bright field microscope, and the sperm heads are stained red and the tails stained green in this NF-PIC stain. Next slide, please. The reagents and supplies used for NF-PIC is shown in this slide. Next slide. The preparation of the stain involves: number one, the nuclear fast stain is a red stain, and this is prepared by dissolving 5 grams of aluminum sulfate in hot water, to which 0.1 gram of nuclear fast red dye is added, and this stain is stable for 6 to 8 months at room temperature. To prepare the picro indigo carmine stain, 150 ml of saturated picric acid solution is added in a glass beaker, to which 0.5 grams of indigo carmine dye is added. This dye is light sensitive and the stain should be prepared and stored in a brown or amber colored bottle. And this stain is also stable for 6 to 8 months. Next slide, please. Once the cytospin slides have been prepared, they are fixed, and the second step is the staining using the nuclear fast stain. This is done for 15 minutes, followed by a quick staining with picro indigo carmine. Next slide, please. Here I would like to show you the video clip showing how the staining is done. The smears are fixed and air dried. And the first step is to add the nuclear fast stain. The slides are placed flat on a slide holder, and the slides are flooded with the nuclear fast stain and stained for 15 minutes. At the end of the staining time, we drain the excess of the stain and allow the slides to be placed vertically And this is followed by the next step, which is the staining with the indigo carmine. The slides are again placed on the slide holder, and we flood the slides with indigo carmine. And this staining is done very briefly for about 30 seconds. At the end of the staining, the excess of the stain is drained, and the slides are next washed with ethanol to remove the excess of the stain. The slides are allowed to air dry, and then they will be cover slipped and ready to be observed under a bright field microscope. Next slide, please. For the microscope setup, we use a bright field 100x objective and a 10x eyepiece. We also use the immersion oil for clear visibility of the stain slide. Next slide. The NF-PIC staining results will include a quality control slide, which is prepared from a normal semen sample and stained along with the NF-PIC slides. The results are reported either as no sperm seen or the sperm are seen, and the image on the right shows how the NF-PIC slide is stained. The sperm head stains pink and the tails are stained green, so it is easy to identify the sperm using this NF-PIC stain. Next slide, please. The quality control for this test is very important, and the quality control is done at two stages. One is during the preparation of the smear, and along with the test sample, we include a blank slide, and the blank slide contains only the 2 drops of saline, and this is used to check for any contamination. The second quality control is used at the time of the NF-PIC staining, and this includes a smear from a normal semen sample to check the quality of the stain. And we perform a visual monthly check for the contamination of the working components. Next slide, please. This test was developed in our lab and validated. And in this slide, I would like to share how the nuclear-fast stain allows to identify the sperm in non-seen samples or samples where no sperm are seen on a routine test. As you can see, the spermatozoa were identified in the samples illustrated on the box to the right, and the positive cells were also identified. And when these were compared with the histological or the clinical diagnosis, there was a good match of finding spermatozoa in 45% of the cells, which did not show any sperm on the initial semen analysis. So this is a test that can be used in an embryology lab to identify and confirm azoospermia in the samples which do not show any sperm on a wet prep. Next slide, please. So the take-home message is that even though the incidence of non-obstructive azoospermia in infertile men is 10%, it cannot be confirmed on a simple routine semen analysis. Our lab has developed a test that confirms the diagnosis of azoospermia in a given sample, and the NF-PIC stain identifies the presence or absence of sperm in the sample. The identification of the sperm on special staining, it allows the clinicians to select azoospermic patients who have the highest chances of finding sperm during the surgical sperm retrieval techniques. Next slide, please. In the end, I would like to share this group picture, and thank you very much for your attention.
Connect with our program by connecting on our LinkedIn, and these are the benefits. Almost every single day, we provide update on our research activities by posting hundreds of research articles freely, completely downloadable, that anybody can read. We also post the latest research which are presented at prestigious meetings such as ACRM, ASHRI, and AUA. You can learn about our virtual research opportunities, which we have just started. You can learn about our online training and opportunities to collaborate with our team. Just go to the LinkedIn and send a request to Ashok Agrawal, and it will from Cleveland, and it will come to us, and we will allow you to connect to our program. This is actually the best way to learn all the advances that are happening.